Chapter 19. The Kidneys. We'll begin by exploring the functions of the kidneys, often referred to as the body's natural filtration units. They do much more than just produce urine. They're key players in regulating electrolytes, balancing fluid levels, and controlling blood pressure. Next, we'll review the anatomy of the urinary system. Here, we'll introduce you to the key components: the kidneys, the ureters, the bladder, and the urethra. Think of them as a carefully coordinated team, each with a specific job to do. After that, we'll dive into an overview of kidney function. This will set the stage for understanding the more detailed processes that follow. From there, we'll break down the journey of fluid through the kidneys, starting with filtration, the process where blood plasma is filtered at the glomerulus, separating waste from what your body needs to keep. But filtration is just the beginning. Next comes reabsorption, where the body reclaims valuable resources like water, glucose, and essential ions from the filtrate, ensuring nothing important is wasted. Then, we'll look at secretion, a process that allows the body to get rid of additional waste products and toxins by actively moving them into the nephron. Once filtration, reabsorption, and secretion are complete, we reach excretion. This is when the final product, urine, leaves the kidneys and makes its way out of the body. Finally, we'll discuss micturition, or the process of urination, which involves the coordination of muscles and nerves to control when and how urine is released. Let's talk about the remarkable functions of the kidneys. These small, bean-shaped organs may not look like much, but they're responsible for maintaining your body's internal stability in more ways than you might think. First up, the regulation of extracellular fluid volume and blood pressure. The kidneys help monitor how much water is retained in the body and adjust blood pressure accordingly. When your body needs more water, they conserve it. When there's too much, they get rid of it through urine. This regulation is critical for preventing dehydration or hypertension. Next, we have the regulation of osmolarity, a fancy term for keeping the concentration of solutes, like sodium and potassium, within precise limits. The kidneys carefully balance water and solutes to ensure cells function properly. Maintenance of ion balance is another key function. Ions like sodium, potassium, and calcium are essential for nerve conduction, muscle contraction, and other vital processes. The kidneys fine-tune their levels, ensuring everything stays in balance. They also play a role in homeostatic regulation of pH. By excreting hydrogen ions and reabsorbing bicarbonate, the kidneys help maintain the body's pH around 7.4, just the right level for optimal enzyme activity in metabolic processes. Of course, one of the kidney's most well-known jobs is the excretion of wastes. They filter out harmful byproducts of metabolism, such as urea, creatinine, and toxins, and eliminate them through urine. Finally, the kidneys contribute to the production of hormones that regulate key functions throughout the body. They produce erythropoietin, which stimulates red blood cell production, and renin, which plays a role in blood pressure regulation. In short, the kidneys are multitasking powerhouses, maintaining homeostasis and protecting your overall health with each heartbeat. Up next, we'll dive deeper into how these processes work at the cellular level. The kidneys are retroperitoneal organs, meaning they sit toward the back of the abdominal cavity, just behind the peritoneum. This unique positioning protects them while still allowing easy access to the blood vessels that supply them. Speaking of blood vessels, it's important to highlight the distinction between renal arteries and renal veins. The renal arteries deliver oxygenated blood to the kidneys for filtration, while the renal veins carry filtered blood away, returning it to the general circulation. At the core of kidney function is the nephron, the functional unit of the kidney. Each kidney contains about 1 million nephrons, and they are the structures responsible for filtering blood, reabsorbing nutrients, and excreting waste. The kidneys themselves are arranged into two distinct layers. The outer cortex, which contains about 80% of the nephrons, referred to as cortical nephrons. The inner medulla, which houses the remaining 20%, These are known as juxtamedullary nephrons and play a key role in concentrating urine. We'll explore this further in the next slide. The vascular system within the kidneys is also fascinating. Blood flow follows a specialized portal system that is unique to this organ. Blood enters through the afferent arteriole and passes into the glomerulus, a tuft of capillaries where filtration begins. From there, it moves into the efferent arteriole and then to the peritubular capillaries or vasa recta, depending on the type of nephron. Let's visualize the inner workings of the kidneys. On the left, you can see a cross-section highlighting the outer cortex and inner medulla. The cortex houses most of the nephrons, while the medulla contains specialized juxtamedullary nephrons that concentrate urine. As the filtrate moves through the nephrons, it flows toward the renal pelvis and exits through the ureters on its way to the bladder. On the right, we see the renal arteries branching into smaller vessels, eventually reaching the afferent arterioles and glomeruli, where blood is filtered. After filtration, blood exits through the efferent arterioles and flows into capillaries that aid in reabsorption and secretion. This system ensures that waste is efficiently removed, while valuable nutrients and water are conserved, keeping your body in balance. Let's walk through the key structures of the nephron, the kidney's filtration powerhouse. It all begins in Bowman's capsule, where plasma from the blood is filtered through the glomerulus, forming the renal corpuscle. From there, the filtrate enters the proximal tubule, the first site of reabsorption, where essential nutrients and water are reclaimed. Next is the loop of Henle, which dives into the medulla and has two distinct sections: the descending limb, which allows water to be reabsorbed, and the ascending limb, where ions like sodium and chloride are recovered. The filtrate then moves to the distal tubule, where further fine-tuning of ion balance and pH occurs. Afterward, it flows into the collecting ducts, which converge and direct the final urine product toward the renal pelvis. Finally, there's the Juxtaglomerular apparatus, a key regulatory structure where the ascending limb passes between the afferent and efferent arterioles. It plays a crucial role in monitoring blood pressure and sodium levels to maintain homeostasis. Together, these tubular elements ensure precise filtration and balance, allowing the kidneys to efficiently process blood and produce urine. Let's begin by looking at the broader urinary system and then zoom in on the structure of the kidneys. On the left, we see an overview of the urinary system. It starts with the paired kidneys, located on either side of the spine. These organs filter blood and produce urine. From there, urine flows down the ureters, thin tubes connecting the kidneys to the urinary bladder, where it is temporarily stored. When it's time to eliminate waste, urine exits through the urethra. On the right, we have a closer look at the kidneys' anatomical positioning. The kidneys sit retroperitoneally, tucked toward the back of the abdomen near the lower ribs. This position protects them while giving access to major blood vessels like the renal artery and renal vein, which ensure a constant supply of blood for filtration and carry filtered blood back to circulation. This layout of the urinary system shows how efficiently the body moves, stores, and excretes waste. Next, we'll dive deeper into how the kidneys function at the microscopic level to regulate your body's fluid balance and maintain homeostasis. Let's take a closer look at the functional and vascular organization of the nephron within the kidney. On the left, we see how the nephron begins in the cortex of the kidney, with Bowman's capsule surrounding the glomerulus to initiate filtration. As the filtrate moves along, it passes through the proximal tubule and descends into the loop of Henle, which dips down into the medulla before ascending back toward the cortex. This layout is crucial, especially for juxtamedullary nephrons, which extend deeper into the medulla. These nephrons play a key role in concentrating urine by reabsorbing water and solutes. In the middle, we see the portal vascular system unique to the kidneys. Blood enters through the afferent arteriole and passes into the glomerulus, where filtration occurs. The filtered blood then exits via the efferent arteriole, but it's not done yet. Instead of leaving the kidney directly, It flows into either the peritubular capillaries, in cortical nephrons, or the vasa recta, in juxtamedullary nephrons. On the right, you'll notice how the vasa recta closely follows the loop of Henle, creating a countercurrent exchange system that helps conserve water and electrolytes. The filtrate, now concentrated and refined, flows through the collecting duct on its way to the renal pelvis. This complex but efficient system ensures that the kidneys can fine-tune the composition of blood and maintain the body's fluid balance with remarkable precision. Let's break down the structure of the nephron and the critical role of the glomerulus in filtration. On the left, you'll see an untwisted diagram of the nephron, allowing us to follow the flow of fluid step by step. It begins in Bowman's capsule, where plasma from the blood is filtered through the capillaries of the glomerulus, forming the initial filtrate. From there, the filtrate travels through the proximal tubule, where essential nutrients like glucose, sodium, and water are reabsorbed back into the bloodstream. Next, it enters the loop of Henle, starting with the descending limb, which allows water to be reabsorbed, followed by the ascending limb, where salts like sodium and chloride are recovered. After this, the filtrate reaches the distal tubule, where further adjustments to ion concentration and pH occur. The final stage is the collecting duct, where the filtrate is fine-tuned before being directed to the bladder as urine. On the right, you'll see an electron microscope image of the glomerulus with its ball-like mass of capillaries. This structure is where the magic of filtration happens, separating water, electrolytes, and small solutes from the blood while retaining larger molecules like proteins. This efficient flow through the nephron and the intricate filtration of the glomerulus work together to maintain balance and homeostasis in the body. The kidneys perform three main tasks to maintain balance in the body: filtration, reabsorption, and secretion. Let's explore each of these essential processes. First, there's filtration, the starting point of urine formation. Here, fluid from the blood is filtered into the lumen of the nephron at the renal corpuscle, which includes Bowman's capsule and the glomerulus. The filtered fluid, called filtrate, contains water, ions, and small solutes. If it's not reabsorbed later in the process, it will be excreted as urine. Next comes reabsorption, where valuable materials in the filtrate, like water, glucose, and electrolytes, are reclaimed and returned to the blood. This process occurs in the proximal tubule, loop of Henle, and distal tubule with the help of the peritubular capillaries. Lastly, there's secretion, where the body actively moves waste products, toxins, and excess ions from the blood into the lumen of the nephron. This ensures that any remaining unwanted substances can be excreted. Like reabsorption, secretion also involves the peritubular capillaries. Together, filtration, reabsorption, and secretion create a precise system for filtering the blood and maintaining the body's internal environment. In the next slides, we'll explore these processes in greater detail. This diagram shows the untwisted pathway of fluid through the nephron, illustrating how the four key processes, filtration, reabsorption, secretion, and excretion, work together to maintain the body's balance. It all begins at the glomerulus in Bowman's capsule, where filtration occurs. Plasma and solutes are filtered from the blood into the nephron, forming the initial filtrate. From here, the filtrate moves through the proximal tubule, where reabsorption of valuable substances like sodium, water, and glucose begins. As the filtrate continues into the loop of Henle, water is reabsorbed in the descending limb, while sodium and chloride ions are reabsorbed in the ascending limb. This step is crucial for concentrating the filtrate and conserving water. Next, in the distal tubule, more fine-tuning occurs. Both reabsorption of ions and secretion of waste products, such as hydrogen and potassium ions, help balance pH and electrolyte levels. These processes rely on the interaction between the nephron and the peritubular capillaries. Finally, the filtrate enters the collecting duct, where further water reabsorption occurs depending on the body's needs. Any remaining waste, along with excess water, is excreted as urine. By the time the filtrate leaves the nephron, it has been carefully filtered, adjusted, and concentrated, ensuring that only necessary waste is excreted while vital substances are conserved. This highly efficient system keeps your body in perfect balance. Let's take a closer look at how solutes move through the nephron and how their fate is determined by filtration, reabsorption, secretion, and excretion. It all starts at the glomerulus, where blood enters through the afferent arteriole and undergoes filtration. Substances like water, electrolytes, and small solutes are filtered into Bowman's capsule to form the initial filtrate. This is represented by F, the amount of solute filtered. As the filtrate moves through the tubule system, some solutes are reabsorbed, meaning they are taken back into the bloodstream via the peritubular capillaries. This reabsorption is crucial for retaining substances like glucose, sodium, and water when needed. This step is represented by R. However, not everything is reabsorbed. Some solutes, like excess ions or toxins, are actively secreted into the nephron from the blood. This process ensures that unwanted substances are added to the filtrate for removal and is represented by S. Finally, the amount of solute excreted in the urine depends on this balance. Amount filtered minus amount reabsorbed plus amount secreted equals amount excreted. This equation demonstrates the kidney's ability to precisely regulate solute excretion and maintain homeostasis. Through this careful balance, the kidneys ensure that waste is eliminated, while valuable substances are conserved. We start with the renal corpuscle, which includes the glomerulus and Bowman's capsule. This is where filtration begins, as mostly protein-free plasma is filtered from the capillaries into the nephron. This process forms the initial filtrate that will undergo further modification. Next, we move into the proximal tubule, the first site of reabsorption. Here, the nephron reclaims organic nutrients like glucose, ions like sodium, and water through isosmotic reabsorption. It's also where unwanted substances like metabolites and xenobiotics, such as penicillin, are secreted into the filtrate for elimination. The filtrate then flows into the loop of Henle, where water and ions are reabsorbed selectively. The descending limb is permeable to water, allowing reabsorption to occur, while the ascending limb reabsorbs ions like sodium and chloride. This countercurrent mechanism helps concentrate the interstitial fluid in the medulla and contributes to the kidney's ability to concentrate urine. Finally, the filtrate reaches the distal nephron, which includes the distal tubule and collecting duct. Here, the nephron fine-tunes salt and water reabsorption based on the body's needs. This segment also plays a critical role in maintaining pH homeostasis, ensuring that blood pH stays within its optimal range. Through this segmented process, the nephron ensures that nutrients are reabsorbed, waste is excreted, and the body maintains a perfect balance of electrolytes and fluids. Let's follow the journey of filtrate as it moves through the nephron and undergoes changes in both volume and osmolarity. It all begins at the renal corpuscle, where 180 liters of filtrate are produced per day. At this stage, the filtrate is nearly identical to plasma, with an osmolarity of about 300 mosm. This initial filtrate contains water, solutes, and small molecules, but the journey through the nephron will modify its composition significantly. In the proximal tubule, about 70% of the filtrate is reabsorbed. Both solutes and water are reclaimed here, reducing the volume of filtrate to 54 liters per day, while the osmolarity remains around 300 mosm due to the proportional reabsorption of water and solutes. As the filtrate reaches the loop of Henle, a unique transformation occurs. The descending limb reabsorbs water, while the ascending limb is responsible for reabsorbing more solutes than water. By the time the filtrate exits the loop, it is highly diluted, with a reduced volume of 18 liters per day and an osmolarity of 100 mosm. In the distal tubule and collecting duct, fine-tuning takes place. Here, reabsorption and secretion adjust the final composition based on the body's hydration status. If the body is dehydrated, more water is reabsorbed, concentrating the urine. The final volume is about 1.5 liters per day, with osmolarity ranging from 100 to 1200 mosm, depending on hydration levels. This dynamic process highlights the nephron's ability to precisely regulate fluid volume and osmolarity, ensuring that the body maintains balance under various conditions. Filtration is the first step in the kidney's process of filtering and purifying blood. But not all the plasma that flows through the kidneys is filtered. Only about 20% of it enters the nephron as filtrate. With a plasma flow rate of 625 milliliters per minute, only 125 milliliters are filtered. This precise filtration ensures that the kidneys can selectively regulate what stays in the blood and what is removed. Filtration occurs at the renal corpuscle, which serves as a filtration barrier, ensuring that only small molecules pass through while larger molecules like proteins remain in the blood. Let's break down these filtration barriers. The first layer is the glomerular capillary endothelium, which contains fenestrated capillaries, tiny pores that allow water and small solutes to pass through while blocking larger components like red blood cells. The glycocalyx, a negatively charged layer, further prevents large proteins from entering the filtrate. Next is the basement membrane, which acts as a physical and electrostatic filter, ensuring that large and negatively charged molecules stay in the bloodstream. The final barrier is the epithelium of Bowman's capsule, which contains specialized cells called podocytes. Their foot processes create narrow filtration slits, allowing only filtered plasma to pass into the lumen of the nephron. These combined barriers ensure that the filtrate entering Bowman's capsule is free of large proteins and cells, but still contains water, electrolytes, glucose, and small solutes ready for processing by the nephron. Up next, we'll explore how this filtrate is refined further. Let's take a closer look at how much plasma is filtered through the nephron and what happens to it along the way. Blood enters the glomerulus through the afferent arteriole, and this plasma volume is considered 100% of the incoming flow. However, only about 20% of this volume is filtered through Bowman's capsule and enters the nephron as filtrate. The remaining 80% of the plasma exits through the efferent arteriole and continues into the peritubular capillaries to support further reabsorption and secretion. Now, what happens to this filtered volume? As it flows through the nephron, more than 99% of the filtered fluid is reabsorbed back into the bloodstream. This reabsorption occurs in the proximal tubule, loop of Henle, distal tubule, and collecting duct, ensuring that only a small amount of the filtrate is lost as urine. By the time the process is complete, less than 1% of the original plasma volume is excreted as urine. This tiny fraction contains waste products like urea, excess ions, and toxins, helping maintain the body's balance. Meanwhile, the reabsorbed fluid, more than 99% of the plasma entering the kidneys, returns to systemic circulation through the peritubular capillaries. This filtration and reabsorption mechanism is a highly efficient system that allows the kidneys to filter vast volumes of blood while conserving essential fluids and solutes. This balance ensures that only the necessary waste leaves the body while vital components remain in circulation. Let's zoom in on the renal corpuscle, where the process of filtration begins. On the left, you see an illustration of the renal corpuscle, which consists of Bowman's capsule and the glomerulus. Blood enters through the afferent arteriole and flows into the glomerulus, a network of capillaries where filtration occurs. Surrounding these capillaries are specialized cells called podocytes, which are part of the capsular epithelium. Podocytes extend finger-like structures known as foot processes, creating narrow filtration slits that only allow small molecules like water, ions, and glucose to pass through. Larger components, such as proteins and blood cells, are retained in the bloodstream and exit through the efferent arteriole. On the right, a micrograph provides a closer look at the intricate structure of the podocyte foot processes wrapping around the glomerular capillaries. These structures play a crucial role in regulating what is filtered, ensuring that only plasma components of the right size make it through to Bowman's capsule. Once filtered, the plasma enters the lumen of Bowman's capsule and is referred to as filtrate. From here, it moves to the proximal tubule, where reabsorption begins. This filtration barrier is highly efficient and selective, protecting the body by preventing the loss of large molecules while allowing waste products and excess fluid to be processed by the nephron. Filtration in the glomerulus isn't just about anatomy. It's driven by the interaction of three key pressures that regulate how much plasma is filtered into Bowman's capsule. The first and most important pressure is capillary blood pressure, which is a type of hydrostatic pressure. This pressure, typically around 55 mmHg, is generated by the flow of blood through the glomerular capillaries. It favors filtration, pushing plasma and solutes out of the capillaries and into Bowman's capsule. However, opposing this outward push are two forces. Capillary colloid osmotic pressure, which is caused by the presence of plasma proteins that cannot be filtered. These proteins pull water back into the capillaries, creating a pressure of about 30 mmHg. This pressure opposes filtration by resisting the movement of fluid into Bowman's capsule. Capsule fluid pressure, the hydrostatic pressure inside Bowman's capsule itself, is typically around 15 mmHg. This also opposes filtration, as the fluid already present in the capsule creates resistance against new filtrate entering the space. When we combine these pressures, we can calculate the net filtration pressure. This small but crucial pressure difference is what drives fluid into the nephron, initiating the filtration process. This delicate balance allows the kidneys to regulate filtration efficiently, ensuring just the right amount of plasma is processed without overwhelming the system. In the next slide, we'll dive into how changes in these pressures can impact kidney function. Let's break down the factors that determine glomerular filtration rate, or GFR, the rate at which plasma is filtered into Bowman's capsule. This process is essential for regulating fluid and electrolyte balance in the body. On the left, we see a diagram showing the calculation of net filtration pressure. As we discussed earlier, this pressure is the balance between three forces. Capillary blood pressure, around 55 mmHg, which pushes fluid out of the capillaries. Colloid osmotic pressure, about 30 mmHg, which opposes filtration by pulling fluid back into the blood due to plasma proteins. Capsule fluid pressure, typically 15 mmHg, exerting resistance from the fluid already inside Bowman's capsule. But GFR isn't determined by pressure alone. It also depends on the filtration coefficient. On the right, we see two key components of this coefficient. Surface area of the filtration slits. The larger the available area for filtration, the higher the GFR. Filtration barrier permeability. How easily substances pass through the filtration barrier. If the barrier is more permeable, GFR increases. Together, the pressure and the filtration coefficient maintain a normal GFR of around 125 ml. If any of these factors change, such as a drop in capillary pressure or damage to the filtration barrier, GFR can decrease, affecting the kidney's ability to filter and regulate fluids. In summary, GFR is a carefully balanced process that ensures the kidneys filter just the right amount of plasma, adapting as needed to maintain homeostasis. The GFR is the amount of fluid filtered by the kidneys per minute, typically around 125 ml. Maintaining a relatively constant GFR is critical to ensure the kidneys filter blood efficiently without overloading or underperforming. The primary mechanism for controlling GFR is through changes in arterial resistance. By adjusting the diameter of the afferent and efferent arterioles, the body can regulate how much blood reaches the glomerulus and how quickly it exits. Here's how it works. Increased resistance in the afferent arteriole restricts blood flow into the glomerulus, decreasing GFR. Increased resistance in the efferent arteriole creates back pressure within the glomerulus, increasing GFR. Decreased resistance in the afferent arteriole allows more blood to enter the glomerulus, increasing GFR. Decreased resistance in the efferent arteriole reduces back pressure, decreasing GFR. These changes allow the body to adapt quickly to fluctuations in blood pressure and maintain a stable filtration rate. This is particularly important during activities like exercise or dehydration, where rapid adjustments in blood flow are necessary to protect kidney function and preserve fluid balance. Let's dive deeper into how changes in arterial resistance directly affect GFR and renal blood flow. In the top diagram, panel C, you see that resistance changes in the afferent and efferent arterioles can alter both the flow of blood into the glomerulus and the pressure driving filtration. This balance ensures the kidneys can adapt quickly to maintain a relatively stable GFR. Let's look at two scenarios. Panel D, increased resistance in the afferent arteriole. When the afferent arteriole constricts, less blood enters the glomerulus, leading to decreased renal blood flow, or RBF, and a drop in capillary hydrostatic pressure, or P. As a result, GFR decreases. This mechanism is often triggered when the body needs to conserve fluids, such as during dehydration. Panel increased resistance in the efferent arteriole. Constriction of the efferent arteriole causes blood to back up in the glomerulus, increasing capillary pressure and enhancing GFR. This can occur when the body needs to filter more plasma, such as after consuming excess fluids. These dynamic changes ensure that GFR remains relatively stable, even when blood pressure fluctuates. This regulation is crucial for protecting kidney function and maintaining fluid and electrolyte balance. In summary, by modulating resistance in the afferent and efferent arterioles, the body fine-tunes GFR based on current physiological demands, preventing over- or underfiltration. One of the kidney's most impressive abilities is their capacity to maintain a relatively constant GFR across a wide range of blood pressures, thanks to a mechanism called autoregulation. In this graph, you can see how GFR remains stable between a mean arterial blood pressure of 80 to 180 mmHg. Within this range, the kidneys automatically adjust the resistance in the afferent and efferent arterioles to ensure that the filtration rate stays close to 180 liters per day. This means that even when blood pressure fluctuates, such as during exercise or rest, the kidneys can maintain consistent filtration. When blood pressure drops below 80 mmHg, GFR decreases significantly. This happens to conserve fluid, reducing urine output and preserving blood volume. On the other hand, if blood pressure rises above 180 mmHg, GFR increases, and the kidneys excrete more fluid to help protect against excess pressure. This autoregulatory mechanism ensures that the kidneys can perform their filtration function without damage, even under varying conditions. It's an essential feature that allows the body to balance fluid and electrolytes and prevent over- or underfiltration during changes in daily activity. The kidneys have built-in mechanisms to regulate GFR, ensuring that it stays stable even when blood pressure fluctuates. Two primary mechanisms, the myogenic response and tubuloglomerular feedback, work together to control GFR. First, let's look at the myogenic response. This is the intrinsic ability of the smooth muscle in the afferent arteriole to respond to changes in pressure. When blood pressure increases, the arteriole constricts, reducing blood flow and preventing an excessive increase in GFR. Conversely, when blood pressure drops, the arteriole dilates, allowing more blood to enter the glomerulus. This response is similar to the autoregulation seen in other systemic arterioles. Next is tubuloglomerular feedback, which is a more fine-tuned mechanism involving paracrine control. It relies on the juxtaglomerular apparatus, a specialized structure that monitors the composition of the filtrate. Within this apparatus, macula densa cells detect the concentration of sodium chloride in the filtrate. If sodium chloride levels are too high, they signal the afferent arteriole to constrict, reducing GFR. Granular cells, also known as juxtaglomerular cells, secrete the enzyme renin. This triggers the renin-angiotensin system, which helps regulate blood pressure and GFR. Finally, hormones and autonomic neurons also influence GFR by altering arterial resistance. For example, sympathetic stimulation during stress can cause vasoconstriction, reducing GFR to conserve fluids. These combined mechanisms allow the kidneys to maintain precise control over filtration, ensuring that the body can adapt to different physiological conditions while protecting renal function. The juxtaglomerular apparatus, or JGA, is a key player in the kidney's ability to regulate blood pressure and GFR through tubuloglomerular feedback. On the left, you can see how the nephron's ascending limb of the loop of Henle loops back to pass between the afferent and efferent arterioles. This strategic positioning is crucial because it allows the JGA to monitor and respond to changes in the filtrate and blood flow. On the right, we see the components of the JGA. The macula densa cells, located in the distal tubule, sense the concentration of sodium chloride in the filtrate. If sodium chloride levels are too high, this signals that GFR is moving too fast and less reabsorption is occurring. The macula densa releases paracrine signals that cause the afferent arteriole to constrict, reducing blood flow into the glomerulus and lowering GFR. The granular cells, also called juxtaglomerular cells, located along the walls of the afferent arteriole, have another important role. When they detect low blood pressure, they release renin, an enzyme that triggers the renin-angiotensin system. This system increases blood pressure and promotes the retention of sodium and water, helping restore homeostasis. Together, the macula densa and granular cells allow the JGA to serve as a highly efficient monitoring and response system. Whether it's slowing down GFR to prevent excessive filtration or increasing blood pressure when needed, the JGA helps the body maintain optimal filtration and fluid balance. In summary, the JGA is a perfect example of how the kidneys precisely regulate themselves to adapt to changing conditions and maintain homeostasis. Let's take a closer look at how tubuloglomerular feedback helps regulate GFR through the juxtaglomerular apparatus, or JGA. This feedback loop is designed to prevent overfiltration when GFR is too high and restore balance when needed. Step 1. GFR increases. When GFR rises, more fluid is filtered into Bowman's capsule and travels through the nephron. Step 2. Flow through the tubule increases. As filtrate moves faster through the nephron, less time is available for reabsorption. Step 3. Flow past the macula densa increases. The macula densa cells, located in the distal tubule, sense the high flow and elevated sodium chloride concentrations in the filtrate. Step 4. Paracrine signals are released from the macula densa to the afferent arteriole. The macula densa triggers the release of chemical signals that act locally on the afferent arteriole. Step 5. The afferent arteriole constricts. In response to the paracrine signals, the afferent arteriole narrows, restricting blood flow into the glomerulus. Step 6. Resistance in the afferent arteriole increases. With the reduced blood flow, the hydrostatic pressure within the glomerulus decreases. Step 7. GFR decreases. As a result of the reduced pressure and blood flow, GFR returns to a more optimal level, preventing excessive filtration and fluid loss. This feedback mechanism demonstrates how the kidneys adjust GFR dynamically to protect against fluctuations in blood pressure or fluid balance. The precise regulation ensures the kidneys don't overfilter during times of increased blood flow, while maintaining their essential filtration role under normal conditions. Reabsorption is one of the key processes that ensures essential substances are retained in the body while waste is excreted. After filtration in the glomerulus, the filtrate entering the nephron contains not only waste, but also valuable substances like water, glucose, ions, and amino acids that the body needs to reclaim. Reabsorption can be either active or passive. Active reabsorption requires energy, often involving transport proteins that move molecules like sodium ions or glucose against their concentration gradients. Passive reabsorption occurs when molecules, such as water, move along their concentration gradient through osmosis or diffusion. The majority of reabsorption takes place in the proximal convoluted tubule, or PCT, where about 70% of the filtrate is reabsorbed. Here, water and solutes are reclaimed efficiently, ensuring that most of the valuable components filtered from the blood are returned to circulation. As we move further through the nephron, additional reabsorption occurs in the loop of Henle, distal tubule, and collecting duct, fine-tuning the body's fluid and electrolyte balance. In the next slide, we'll explore the mechanisms that drive this process and their role in maintaining homeostasis. Let's take a closer look at the mechanisms that drive tubular reabsorption, a process that allows the body to reclaim valuable substances from the filtrate and return them to the bloodstream. The reabsorption process can occur through transcellular transport, where solutes pass through epithelial cells, or via the paracellular pathway, where they pass between cells through tight junctions. Here's a step-by-step breakdown of how this works. Step 1. Sodium ions are reabsorbed by active transport. Na is one of the key ions involved in reabsorption. Specialized transport proteins actively move Na out of the tubular lumen into the epithelial cells and then into the extracellular fluid. This active transport is a driving force for the reabsorption of many other substances. Step 2. An electrochemical gradient drives the reabsorption of anions. As Na is actively reabsorbed, it creates an electrical gradient that attracts negatively charged ions, such as chloride, promoting their movement into the extracellular fluid. Step 3. Water follows by osmosis. The movement of Na and other solutes lowers the solute concentration in the tubular lumen, creating an osmotic gradient. Water naturally follows the solutes through osmosis, ensuring that the body reabsorbs most of the filtered water. Step 4. Permeable solutes, like potassium, calcium, and urea, are reabsorbed by diffusion. As water is reabsorbed, the concentration of these solutes in the tubular lumen increases, driving their passive diffusion either through membrane transporters or the paracellular pathway. This coordinated system ensures that the majority of the filtrate's valuable contents are efficiently reclaimed, preventing excessive loss of water and electrolytes. The fine-tuning of these processes helps maintain fluid and electrolyte balance in the body. In the upcoming slides, we'll explore how specific hormones influence these reabsorption mechanisms. Sodium plays a crucial role in the reabsorption of many solutes, including glucose, amino acids, and other nutrients. This process primarily occurs in the proximal tubule through sodium-linked cotransport. Here's a step-by-step look at how this mechanism works. Step 1. Sodium moves down its electrochemical gradient. Na concentrations are higher in the tubular lumen than inside the proximal tubule cells. As sodium moves passively into the cell, it uses the sodium-glucose-linked transporter to pull glucose into the cell against glucose's concentration gradient. This cotransport mechanism ensures that even low levels of glucose in the filtrate can be efficiently reabsorbed. Step 2. Glucose diffuses out of the cell into the interstitial fluid. Once inside the cell, glucose moves down its concentration gradient and exits through the glut transporter on the basolateral membrane. From there, glucose is absorbed into nearby capillaries and returned to the bloodstream. Step 3. Sodium is actively pumped out of the cell. To maintain the low intracellular sodium concentration needed for continuous cotransport, sodium is actively pumped out of the cell by the sodium-potassium ATPase pump on the basolateral membrane. This pump uses ATP to move sodium out of the cell and bring potassium in, ensuring the concentration gradient is preserved. This sodium-linked transport system is highly efficient, allowing for the reabsorption of most of the filtered glucose and other essential solutes. Without this mechanism, valuable nutrients would be lost in the urine, leading to imbalances and deficiencies. Renal transport processes are efficient, but they have their limits. Let's discuss what happens when those limits are reached and how this can affect kidney function. Saturation occurs when all transport proteins are fully occupied by the substance they're moving. For example, in sodium-linked glucose reabsorption, if the concentration of glucose in the filtrate is too high, the transporters become saturated and no more glucose can be reabsorbed. This results in excess glucose being excreted in the urine. The point at which this happens is determined by the renal threshold, the plasma concentration at which a substance first begins to appear in the urine. For glucose, the renal threshold typically corresponds to a plasma glucose level of around 180 mg/dl. When this threshold is exceeded, the result is glucosuria or glycosuria, the presence of glucose in the urine. This is commonly seen in individuals with diabetes mellitus, where high blood glucose levels overwhelm the kidney's ability to reabsorb glucose. The excess glucose in the filtrate draws water into the tubule through osmosis, leading to increased urine output, a hallmark symptom of uncontrolled diabetes. Understanding saturation and renal thresholds is crucial for diagnosing and managing conditions that affect kidney function and fluid balance. In the next slides, we'll examine how this concept extends to other solutes and electrolytes. Secretion is an essential process in the nephron that helps the body eliminate waste products and maintain proper homeostasis. Unlike reabsorption, which moves valuable substances back into the bloodstream, secretion actively transports molecules from the extracellular fluid into the nephron's lumen. This process is particularly important in the regulation of key ions, such as potassium. Excess potassium is secreted into the nephron to maintain proper blood potassium levels, which are critical for nerve conduction and muscle contraction. Hydrogen ions. Secreting hydrogen helps regulate the body's pH balance, ensuring that blood pH remains within its normal range of around 7.4. In addition to ions, the kidneys also secrete metabolites, such as byproducts of cellular metabolism, drugs and medications that need to be cleared from the body, toxins that could otherwise accumulate and cause harm. This selective secretion occurs primarily in the distal tubule and collecting duct, where the fine-tuning of urine composition happens. It works hand in hand with reabsorption to maintain the body's fluid, electrolyte, and pH balance. By efficiently removing waste products and excess ions, secretion ensures that only the right balance of solutes remains in the blood, while unwanted materials are excreted as urine. Excretion is the final step of kidney function, representing the net result of filtration, reabsorption, and secretion. It's the process by which waste products and excess substances are removed from the body in the form of urine. We can express this using the equation: excretion equal filtration, reabsorption plus secretion. Understanding excretion is important for assessing kidney function, and one key way to do this is through a concept called clearance. Clearance is a non-invasive way to measure GFR, an indicator of how well the kidneys are filtering blood. Clearance is expressed as the volume of plasma in ml that is completely cleared of a particular substance within a given time, usually 1 minute. It's important to note that clearance doesn't tell us how much of the substance is excreted, but rather how effectively the plasma passing through the glomerulus has been cleared of that substance. To measure GFR, two substances are commonly used. Inulin is a plant-derived polysaccharide that is freely filtered at the glomerulus, but neither reabsorbed nor secreted. This makes it an ideal marker for GFR measurement. However, it's rarely used clinically because it's impractical and requires intravenous administration. More commonly, creatinine is used to estimate GFR. Creatinine is a byproduct of muscle and is constantly produced and excreted. Although a small amount of creatinine is secreted in the urine, making the measurement slightly less accurate, it's widely used because it is a non-invasive test that relies on simple blood and urine samples. The test is known as creatinine clearance. By measuring clearance, clinicians can assess how well the kidneys are functioning and detect any potential signs of renal impairment, allowing for early intervention when necessary. The process of micturition, or urination, is the final step in the urinary system's role of removing waste from the body. Once urine leaves the collecting ducts, its composition does not change. It is collected and stored in the urinary bladder until the body signals that it's time to void it. Let's break down how this process works. Urine flow is controlled by two key sphincters. The internal sphincter, made of smooth muscle, is under involuntary control. The external sphincter, made of skeletal muscle, is under voluntary control, allowing us to consciously delay urination when necessary. The initiation of micturition begins with a simple spinal reflex. As the bladder fills, stretch receptors in the bladder wall detect the increasing volume and send signals to the spinal cord. These signals trigger parasympathetic fibers to stimulate the contraction of the bladder's smooth muscle, increasing pressure within the bladder. As pressure rises, the internal sphincter automatically opens, allowing urine to move toward the external sphincter. At the same time, somatic motor neurons controlling the external sphincter are inhibited, promoting its relaxation and allowing urine to be expelled. Although this reflex is automatic, the brainstem and cerebral cortex can override it. For example, during toilet training or when holding urine, the external sphincter remains contracted under conscious control until it's appropriate to urinate. This coordination between reflexes and voluntary control ensures that micturition occurs at the right time, preventing accidental leakage while efficiently eliminating waste. Micturition, or urination, is a carefully coordinated process that involves both reflexive control and voluntary input from the brain. Let's walk through how this system transitions from bladder filling to emptying. Panel A. Bladder at rest. When the bladder is relaxed and filling with urine. The bladder's smooth muscle is in a relaxed state, allowing it to expand and hold urine. The internal sphincter, or smooth muscle, remains passively contracted, preventing urine from flowing out. The external sphincter, or skeletal muscle, is actively contracted under conscious control, keeping urine inside. Tonic signals from the brainstem motor neurons maintain this state of contraction. But as the bladder fills and stretch receptors are activated, the system prepares to transition to the process of micturition. Panel B. Micturition process. As the bladder fills, its walls stretch, and sensory neurons send signals to the spinal cord. In response, parasympathetic neurons fire, causing the bladder's smooth muscle to contract. This increases pressure inside the bladder. Simultaneously, motor neuron signals to the external sphincter are inhibited, allowing it to relax. The internal sphincter is passively pulled open due to the pressure increase, and the external sphincter relaxes, allowing urine to exit the body. The brainstem and cerebral cortex can influence this reflex. For example, during toilet training or social situations, the brain can inhibit the reflex by maintaining the external sphincter's contraction until urination is appropriate. Alternatively, higher brain input can facilitate micturition by promoting the relaxation of the external sphincter when desired. This integration of reflexive and voluntary control ensures that urination occurs efficiently while allowing for conscious control when necessary. The urinary system is a story of balance and precision. It starts with the kidneys, small but powerful organs working tirelessly to filter blood, regulate electrolytes, control pH, and maintain blood pressure, all while removing waste. We explored the anatomy of this system, from the kidneys to the ureters, bladder, and urethra, each working together to collect, store, and eliminate urine. The kidneys take the lead, using filtration to separate waste and excess fluid from the blood. From there, reabsorption ensures valuable substances like water, sodium, and nutrients are reclaimed, while secretion actively eliminates toxins, excess ions, and drugs. All of this leads to excretion, where the filtered waste exits the body as urine. The final act is micturition, a beautifully coordinated process involving reflexes and brain control, allowing us to release urine at the right time. Together, these processes demonstrate how the urinary system quietly and efficiently keeps our body in balance, working around the clock to ensure survival. And most of the time, we don't even notice it.